There are different types of transplants, of course, that are performed clinically. Some of those are organ transplants, so heart, lung, liver, kidney, pancreas, intestinal transplants.
There's a variety of kinds of transplantation. It ranges from the cell transplantation, which we're very familiar with in terms of bone marrow transplants or stem cell transplants, or even something as simple as blood transfusions, which is really a cell transplant.
One of the major problems in organ transplantation at the moment that has persisted for decades is that we have a critical shortage of donors. One of the ways that we could address the chronic organ donor shortage is by allowing individuals who need organs to receive organs from individuals of incompatible blood types.
When we worry about solid organ transplantation, such as kidney or heart transplantation, we are worried about the organ being rejected by the person receiving it.
Glycomics impacts transplantation because some of the structures on the organs and cells that we transplant are glycan structures. And if we mismatch these, or if we have incompatibilities between donors and recipients in these glycan structures, there can be a very destructive immune response which will place the patient in danger and potentially destroy the transplant. When we think about transplantation worldwide, we know that at a minimum, probably 2 million people per year would benefit from an organ or cell transplant. This research that we're doing would have an impact on improving access to transplantation for many of these people because we could manage safely the incompatibilities in the glycan structures between the donor and the recipient. So this is really novel work that isn't being done and hasn't really been looked at since the days that Karl Landsteiner described the major blood group system in the early 1900s.
Well, the research we're doing, if we can really understand how a baby or how hematopoietic stem cell transplantation has altered the very major rejection of a solid organ, a heart transplant, we could make the organ available to anybody.
Some of the challenges with the transplantation involve the storage of the cells, the tissues, or the organs outside of the human body. So when we take cells, tissues, or organs outside the human body, they start to undergo a decay. They start to degrade in quality because the body has a natural repair process that repairs the tissues or the cells in situ when they're actually in the body. Our spin-off company, PanTHERA CryoSolutions, allows us to start to incorporate it into products that we want to extend the storage or improve the quality during storage. And through the research that we've done and now through the translation of that research, we're able to bring that to Canadians. By allowing us to protect the cells or the tissues or the organs during low temperature storage, it allows us to extend the time and ultimately the distance that we have in order to actually recover a donated cell or tissue or organ and then transplant it into a recipient.
The colleagues in chemistry in particular, with whom clinical immunologists and physicians and surgeons may not have the ability to generate those interactions very easily. The GlycoNet framework allows us to do that and encourages us to do that and enables us to do that.
GlycoNet's early on provided the funding, which was really critical for research. Without that funding, researchers have a very difficult time exploring some of these concepts and developing some of these ideas. To develop this technology and actually get it into the clinic where we actually can start to trial it in different model systems. At a very critical time for us to actually do what we've done over the last few years.